Question number 126. Name the plant growth regulator which upon spraying on sugarcane crop increases the length of the stem, thus increases the yield of sugarcane crop. First option is abscisic acid, second option cytokinins, third option gibberellin, fourth option ethylene. We know that abscisic acid is responsible for seed dormancy, that is growth inhibiting hormone. Cytokinins promotes cell division, and gibberellins are useful for internodal elongation. So spraying of gibberellins in case of sugarcane is useful for increasing the length of the stem as well as crop yield is also. And ethylene is a fruit ripening hormone. That means the right option for the given question number 126 is third option. And next one is question number 129. If you observe 129, which of the following pair is of unicellular algae? They are asking about the unicellular algae. And the first option, chlorella and spirulina. Second option, laminaria and sargassum. And third option, gracilaria and giladium. And fourth option is anabina and volvax. If you observe in all these four options, the first option is the right option. They are, both of them are unicellular algae, and they are used as a rich source of proteins, as a single cell proteins, chlorella and spirulina. Even astronauts, space travelers are using this chlorella and spirulina as a source of food materials, as a single cell proteins. The right option for question number 129 is first option. Question number 130. If you observe question number 130, which of the following statements about inclusion bodies is incorrect? Students, you must be very careful. You have to observe that they are asking about incorrect statements. And here the first option, these represents reserve materials in cytoplasm. Yes, inclusion bodies are present in the form of synophysin granules or phosphate granules or glycogen granules. That means all these are the reserve food materials present in the cytoplasm of prokaryotic cells, and they are not bounded by any membrane. Yes, membrane is absent. These are involved in ingestion of food particles. So these are not involved in ingestion of food particles. They don't have any role in ingestion of food particles, and they lie free in the cytoplasm. That is also right. That means the statement which is not related to question number 130 is third option. And question number 137, next question is, in light reaction, plastoquinone facilitates the transfer of electrons from, see here, very important point, especially they are given options, photosystem, first option, photosystem 1 to ATP synthase, second option, photosystem 2 cytochrome B6F complex, and third option is cytochrome B6F complex to photosystem 1, and fourth option is photosystem 1 to NADP. And let us see once how the non-cyclic electron transport occurs. During non-cyclic electron transport, this is photosystem 2 reaction center, and photolysis of water occurs nearby this photosystem 2, that releases 2 protons, 2 electrons and half O2. That means the electron deficiency in photosystem 2 is always filled by breakdown of water molecule. These excited electrons are received by primary electron acceptor, that is pheophytin. From pheophytin, the electrons are transferred to plastoquinone. From plastoquinone, these electrons are transferred to a multi-protein complex, that is cytochrome B6F complex. From that, these are transferred to next electron carrier, a mobile electron carrier, copper containing electron carrier, that is plastocyanin, and that donates electrons to the deficiency, electron deficiency in photosystem 1 is fulfilled by plastocyanin. From that, electrons are excited. Those are received by the primary electron acceptor A0. From that, finally, the electrons are transferred to the terminal electron acceptor NADP plus, that is reduced into NADP H plus, H plus. This is non-cyclic electron transport. Here, this PQ is involved in the transport of electrons from photosystem 2 to cytochrome B6F. That means the right option for question number 137 is second option. Students, next question 141. If you observe, the ovary is half inferior in. Half inferior means in this type of flowers, thalamus is concave or saucer shaped. Concave or saucer shaped, and ovary is present in the center of the thalamus. Half of the ovary is embedded inside the ovary, and half of the ovary is above the thalamus, and half of the ovary is inside the thalamus. That means half inferior, or we can call it as half superior. This type of ovary is present in peach, plum and rose. That means the right option for question number 141 is first option. And next one is question number 142. If you observe, in case of 142, the process of growth is maximum during they are asking about growth curve, that is sigmoid curve or S-shaped curve. We know that during this growth curve, we can observe the growth in the initial stage. Initial stage we can call it as lag, and later on the growth becomes very high. That stage we can call it as log phase, and third one is senescent phase. That means growth is very rapid within a short period of time, very rapid growth. This we can call it as exponential phase or log phase. During this stage, if all the conditions, suitable conditions are provided, the growth is very rapid. Even in case of bioreactor, during fermentation process, during production of alcoholic beverages, when the favorable conditions are maintained, required temperatures and conditions are maintained, microorganisms are showing log phase growth or exponential growth. That is why the growth is very rapid in this stage. That means the right option for question number 142 is second option. Question number 143. Which of the following is not an inhibitory substance governing the seed dormancy? They are given some of the substances, chemical compounds which are responsible for seed dormancy. First option, para-ascorbic acid, and 2, gibberellic acid and abscisic acid. Fourth option, and phenolic acid. Out of these four, we know that para-ascorbic acid and abscisic acid and phenolic acid are responsible for seed dormancy. And what is about gibberellins? Gibberellins are promoting seed germination, because gibberellic acid, during seed germination, gibberellic acids are produced in embryonal axis, and from these gibberellic acids are transported to allural layer. In allural layer, they are inducing the synthesis of alpha amylase enzyme. That alpha amylase enzyme is a starch hydrolyzing enzyme. That starch hydrolyzing enzyme transported to the endosperm. In endosperm, the starch gets hydrolyzed and produces carbohydrates. These carbohydrates are involving in respiratory process and produces ATP energy. That ATP energy is utilized for cell division process. By that, the length of the embryonal axis will increase, and the increasing length of the embryonal axis causes pressure on seed coat. By that, the seed coat will rupture and seed will germinate. That is the mechanism involved in seed germination, and the phytohormone which promotes seed germination is gibberellic acid. That means it is not responsible for seed dormancy. The right option for given question number 143 is the second option, gibberellic acid. And question number 144, and the oxygenation activity of Rubisco enzyme at photorespiration leads to the formation. This is also very important point we can discuss here. During oxygenation activity, I will represent here, that is RUBP. It is a 5-carbon compound, and Rubisco is having Rubisco shows both carboxylation and oxygenation. If carboxylation occurs, C3 cycle, Calvin cycle occurs. If oxygenation occurs, if it is acting as oxygenating enzyme, photorespiration or glycolate cycle, or we can call it as a C2 cycle will occur. During this process, this produces one molecule of a 3-carbon compound, that is PGA, phosphoglyceric acid or phosphoglycerate, and one molecule of 2 carbon compound, that is phosphoglycolate and phosphoglycerate. That means these two are produced, and see here, one molecule of 3 carbon compound and one molecule of 2 carbon compound is produced. And here in the given options, if you are observed, first option is one molecule of 4 carbon compound and one molecule of 2 carbon compound. That is not right. Second option is 2 molecules of 3 carbon compound. That is also not related. Third option is one molecule of 3 carbon compound. That is somewhat related. And fourth option is one molecule of 6 carbon compound. That means even though there is no correct answer in the question, but among the following answers, the right option is third option, that is one molecule of 3 carbon compound phosphoglyceric acid is produced. That is why the right option for question number 144 is third option.